computer start. Let's see what you need to know here, wrapping up this story with the fats, right, with the lipids. And what are the parts that you need to know? I know lipids can be a bit painful or literally, right? And it can be a bit confusing. But let's focus here on what you should know about the lipids. And then we're going to go into membrane function, and that should be a lot easier and more intuitive. So we had stopped here last time talking about sterols, right? So here there are several terms that sometimes also can be a bit confusing. So what you should know here about the sterols. So basically, they have several subgroups. So you have isoprenes and isoprenols and other types of structures, but mainly when you're talking about isoprenoids, you are talking about lipids that are made up from this 5-carbon backbone that is called isoprene. Okay, so here's the structure of isoprene, and it is 5 carbons. Okay, 1, 2, 3, 4, 5. And what happens is that, so this is like a basic building block of several lipids. And what happens is that you can put this isoprene in chains, or you can fold them in certain type of ring structures, and they give rise to several very, very important lipids. So remember that in order to be a lipid, you need to have a molecule that is mainly carbon and hydrogen. So here you have this type of backbone structure of a sterol, okay? And you see mainly it is carbon and hydrogen. Then you're going to say, oh, there is a hydroxyl group over there. Yeah, but that hydroxyl group is, first, is not charged, and second, it is very small compared to the whole size of this hydrophobic molecule. Okay, so what do you see when you look at this sterol structure? You're going to see 4 rings. 3 of them are have 6 sides. Okay, so you have A, B, C have 6 sides, and one of them has 5 sides. So that's a basic sterol structure. Okay. With this basic structure, you can form molecules such as cholesterol, that is showing out here. So cholesterol has that hydrophilic hydroxyl group, and cholesterol also has this side chain, which is very hydrophobic, that is connected to that carbon over there, which I think is something like carbon 17, but you know, if I don't remember, you don't have to remember. But remember, there is this huge side chain right there on cholesterol. And it is important for cholesterol to have this very hydrophobic side chain, because this huge hydrophobic part allows cholesterol to be inserted in the structure of lipid bilayers. Okay? So here, looking again at this basic sterol structure, you can have not only cholesterol, but you have other types of molecules like phytosterols, and these are mainly occurring in plants. Okay? Zoosterol are animal steroids, so cholesterol would also follow into this animal sterols type of subgroup. So cholesterol is very, very important component of membranes. So besides cholesterol, you also have other forms of steroids that are also very important for life. Like, for example, the fat-soluble vitamins that are derived from steroids, from sterol, and also several hormones, including androgens and progesterone, those types, and testosterone. Okay, the steroid hormones are based on this structure. So here, when you're looking at sterols, you have those isoprenoids. Examples are the soluble, fat-soluble vitamins, right? So here you have this structure showing vitamin A. Okay, so you see here, you don't see the sterol basic structure with the 5 rings, but this thing is made up of that basic isoprenyl 5 carbon structure. Okay, so here you have also an example of vitamin K, right? And here you have 2 forms of vitamin D. So one is the active and the other one is the inactive form. But just having a basic idea of what those vitamins look like and what are they derived from, that's the main idea here. So here, when you're looking at the steroids, right? So you have those 3 6 size rings, carbon rings, and then one that is 5 sides, and yeah, it was carbon 17. And they have this side group over here. And remember that within this sterol structure, you also have those methyl groups that are part of the whole structure. Now, as I mentioned here, cholesterol is very, very important for the body. And a testament to that is that we have 3 different sources for cholesterol. So first, we can eat it, so it can be in the diet. Okay? Second source is that we can synthesize it ourselves, so there are enzymes that synthesize cholesterol. And actually, one of those enzymes is a target of several cholesterol-lowering drugs, right? But we're not going to get into, cholesterol-lowering drugs. But those are the statin drugs. They will inhibit the synthesis of cholesterol, right? And the third source that we have is that we can store cholesterol basically everywhere in the body, and also keeping in mind that cholesterol can be stored even in the membranes, okay, in our cell membrane. Okay. So here, the fatty acid part of the cholesterol can be stored within the membrane, right? So the hydroxyl part would be associating with those phosphate groups of phospholipids, and then the hydrophobic part, it can be inserted inside of the membrane. So here, more of this idea of isoprenoids. They are building up from a 5-carbon basic isoprene, right? So here is again is your isoprene monomer, okay? Now look at this here. If you put this is only one isoprene molecule, and you are adding phosphates to this isoprene molecule, you can end up with some type of active lipid. And you do have other, variations or isotypes of that. Now look at this. If you put 2 of those isoprene structures together, you end up with this other lipid that is called geranyl pyrophosphate. So 2 phosphates in geranyl. If you put 3 of those subunits, you're going to end up with something else. This is farnesyl pyrophosphate. Okay, so you have like 15 carbons. And if you put 2 of those together, you end up with a very, very long hydrophobic molecule that is called squalene. Okay. And this is interesting that it was found in sharks. So this is your squalene molecule. So another view here of isoprenoid molecule is beta-carotene. So this is a structure for beta-carotene. Okay, so it's occurring in vegetables. So basically you have here 8 isoprene units that get grouped together and then modified at the end. All right. So if you put 2 of those of 15 carbon, okay, you get this squalene molecule. That's your 30 carbon. So what you're doing here, you're basically using Lego blocks of 5 carbons to build bigger and bigger molecules. All right. And a side note here for other forms of sterols, and this is called terpenoids. Okay, so the ones before that don't have that the hydroxyl group is called terpenes. So these here are terpenoids. Okay, so we have here comparison of terpenes to terpenoids, and this is just FYI. I'm not going to ask you about terpenoids. But it's interesting to know that like menthol, for example, is a derivative of 2 isoprene units. Okay? And everybody's familiar with menthol. Right? What do you use menthol for? Anybody ever use menthol? Yeah, yeah. What do you use it for? Huh? Cough drops, yeah, they do have some menthol, right? But you have also some creams that you can use for muscle pain. And you say, well, does it make sense? Well, this is very hydrophobic, so it will go through the skin, it will be absorbed, right? And it can help somehow, it relieves a bit of pain. Okay. So there are several uses for menthol. Okay. So here, how do you make cholesterol? So this is something that I would like you to know. Okay, you get this huge, squiggly, big molecule of 30 carbons, and that is your squalene. Okay, and you start to fold it. So this is the same squalene, but it's folded in a different way. And then you start to form the rings, and you end up with this structure for cholesterol ester. Okay, so cholesterol can also have side chains at the hydroxyl side, not only at the carbon 17. So you get squalene, folded, make the rings, and you end up with cholesterol. So how is it that cholesterol is helping with some membrane function? So as you see here, cholesterol is going to be put in the middle of your membrane. So here you see only one leaflet, one side of the membrane, so the other side is here. Okay, so the cholesterol is like right in the middle between 2 big fatty acids that have the here is showing this polar head, but you know those can be phosphates, right? So here you have cholesterol that is like right in the middle. Now, what is it the cholesterol is doing to the membrane? It is really changing how the membrane is moving around in a 2D orientation. Okay, so that is called the fluidity of the membrane. But remember that when we have looked at the lipids before, we have seen something that has to do with how hydrophobic those side chains are, and also we're looking at saturation. For example, if you look at this side chain right here, is it saturated or is it unsaturated? Saturated, right? You don't see double bonds here. This is a straight out line of saturated fatty acid. And we have seen that the more carbons we have in this chain, the higher is going to be the melting point. But if you put a double bond here, then you're going to lower the melting point. Now, what is cholesterol doing here? Cholesterol is not interfering with the melting point. Okay, so cholesterol does nothing for the melting point of the membrane. But what it does is that it's going to make the membrane more fluid. Okay, so it's going to change the fluidity of the membrane and not the melting point. So here, if you are plotting melting point, and here would be a state of the membrane, whether is it solid-like or fluid-like. When you add cholesterol, you are going to get the same melting point, but a shift in the fluidity. You're going to make this membrane more fluid. Okay? So here is an experiment that shows you what it is that this fluidity means. Okay? So basically what you do here, you have a cell. So you have a cell, and you get this little patch on this cell, that's right here, that you are going to observe lipid label, lipids that were labeled with some form of fluorophore. So all this cell has this fluorophore that is shining red and is distributed all over the membrane. Okay, so here's a little 2D interpretation of that part. So this 2D thing is this side right here. So you see here the membrane that has the red label. Okay, so what you do here, you get a laser and you aim at the center of that membrane. So here's the laser. Okay, it's right there. It pulls out the center of the membrane and you burn out those fluorescent markers. All right, so now this is why you have the dark center right there. The laser burned out the thing and they all became dark. So it's not shining, right? So now you start looking at, still keep looking at this area, and you want to count the fluorescence of those particles. So here, look, this first stretch over here that says A has to do with the amount of fluorescence that you started with your membrane that was intact, only with your shiny red parts, okay? So you had like relatively one of a measurement. Then you buzz out here this area. So now your whole square here is going to have lower fluorescence. So after you burned it out with the laser, your fluorescence really went down. Okay. Now you wait and you keep on looking through the microscope. You keep on measuring the fluorescence of this little square that is this little part of the membrane. Okay, so you notice that after some time has passed, you are going to recover some of that fluorescence. So this thing was really dark and now it starts to be not so dark because the lipids can have lateral motion, and this lateral motion that you call the fluidity is going to disperse and basically allow diffusion of those darkened particles throughout the membrane. So after certain time, enough time has passed, all those little dark particles have distributed equally throughout the whole cell, because this is a live cell, okay? And if you look at the center here, you can't find your dark dot anymore because everything just got mixed up by this lateral motion of those phospholipids, okay? So this is what is meant by fluidity. So now when you look after a certain time, you're going to find that everything is uniform, but the total fluorescence is a little bit less than the fluorescence you started with. Because the lipids that were burned out, they're not going to start shining again. So they just dispersed through the membrane. Is that an experiment that is clear, easy to understand? Yeah, everybody got it? Yeah. And here comes your first question. With a level of confidence. Okay. Which of the following is true regarding cholesterol? Yeah. So I don't think you need more than 30 seconds to answer that. Does anybody need more time? Anybody need time? Everybody answered? Okay, so we have people changing their minds. Okay, I'll allow you to change your mind. Up to a minute. Okay. Everybody else change their minds? Okay, I'm going to stop it. What is your answer? It is A. Just mentioned, it changes membrane fluidity. If you have this molecule by itself not interacting with each other, then you're going to have to increase increase the organization of the water molecule around that. Okay? And that goes towards decrease in entropy. Okay? By having this association, you are minimizing the decrease in the entropy, because still you are going to have water molecules that get organized all around the molecule. So just by the lipid being there, you end up with some decrease in entropy, but you want to minimize that. Okay? Everybody got that? It's a bit tricky on these words. Yeah. Yeah, pretty much. Yeah. Okay. So here, how is it that you form membranes or micelles? So here, if you look at this thing that is called a micelle, this is actually the basic structure of soaps. You see that one part is hydrophilic, and you have only one little fatty acid that is connected to that big head over there. Okay? So when you get this type of molecules, these types in water, then the same thing is going to happen. Water is going to push it around, so the hydrophobic part is going to interact with each other, and the hydrophilic part is going to be interacting with the water. But because this molecule has only one fatty acid, it does not allow for enough space for a bilayer to form. Okay, so you have to look also at the geometry of those fatty acids, right? So here, if you have only one tail that is a fatty acid, you are really going to form this micelle. There is only one layer, and you don't have the other layer for a bilayer here. Okay. On the other hand, if you have these two tails, like in phospholipids, okay, now this thing has mainly a boxy or a rectangular type of shape, and that is going to allow this bilayer formation, okay, as you see over here. Okay. So this is the type of structure that you find, of course, in cells, cell membranes, but also in lipid vesicles. Okay, those can be liposomes, microvesicles, exosomes, outer membrane vesicles, bacterial membrane vesicles. Everything is going to have this triangular shape. So here's a little picture showing you how to make fatty acids. Okay, so basically you have your hydrophilic part, and here's your lipid tail. Now, if you use a strong base that steals the hydrogen, you end up making salt out of the fatty acid, and this is basically the principle for soaps. So soaps, when you use soaps to take a shower, wash your hands, wash your hair, the micelles in the soap, they trap organic fat-soluble particles inside of them. That's how soap is actually effective. So here is this basic idea of the geometrical shape of your fatty acids. Okay, so you have here, if you have only one fatty acid tail, so you have this more or less triangular shape, you can't really form your bilayer. So you form the micelles. So with your two tails, then it is okay to form the bilayer. Now, if you have three tails, what is an example of this thing that has three tails? Like triacylglycerol. Right? So with three tails, you can't really form the bilayer. It's not in the proper geometric conformation. So interesting things about these vesicles. So we have liposomes, we have exosomes, microvesicles. So basically they can all form spontaneously. Okay? What is it that's going to make the bilayer? You're going to have glycolipids. Those are lipids that have some sugar molecules mainly on the outside. Okay, you're going to have glycerophospholipids, right? So glycerol, and you're going to have also sphingolipids. Those are your sugars that are more complex. Remember the sphingolipids? You have what? Cerebroside, ganglioside. Those are your sphingolipids, right? So here, when you look at the formation of those natural membranous structures, you also see these structures that are called exosomes and microvesicles. And look at this. Every living cell is capable of making those microvesicles and exosomes. And those are just budding out of the membrane. Okay? And what is it that they carry? They can carry a lot of stuff. They can carry mRNA or small interfering RNA, siRNAs. Okay, so mRNA, miRNA. They can carry some proteins, okay? And they can really carry a lot of stuff that serves as communication between one cell and another cell. And there's a lot of science coming up about this cell communication mode, okay? So the difference here between microvesicles and exosomes is that microvesicles tend to just bud out of the membrane and they are bigger. Exosomes, they tend to be formed inside of the cell and then be exocytosed out through the membrane, and they're a lot smaller. Okay. So here is a clinical application for the function of those exosomes, which can also be called nanoparticles, right? So here you have examples of nucleic acid vaccines. Right? So everybody got vaccinated against the COVID, right? So that was the first implemented large-scale usage of liposomes in nucleic acid vaccines. Right? So what is happening here with your COVID vaccine? You had actually two types. You had one type that was using DNA, right? So there was a type that would use a modified adenovirus. So that was the Johnson & Johnson, right? And you had this other type that was the more used one. That was the, what's the name of the other? Pfizer, yeah, Moderna, Pfizer, and there's still another one, AstraZeneca. That could be also DNA. So what happens here? With this Pfizer vaccine, you had a piece of RNA that got packed in this nanoparticle that is a membrane enclosed thing, okay? So that thing gets into the injection, right? Injects in your body, okay? Now, where was it you got the injection? In the muscle, right? So then what happens? This particle fused with a cell membrane that happens to be the first cell that was close to the injection site, which happened to be your muscle, right? So it fused with the cell membrane, delivered the messenger RNA to the cytoplasm. In the cytoplasm, you have the machinery to make your viral proteins that were encoded in that messenger RNA. These viral proteins got presented on the surface of the cell, and then you started to teach your immune system cell to respond to that viral protein, because that viral protein is not part of yourself, is not self protein, okay? So immune system came there, looked at that protein and said, something is wrong with this cell. I think it is infected. Let me gather all of my immune system partner cells and start making antibodies against this virus protein, because most likely other cells are infected too. Okay? So this is the principle of how your Moderna vaccine worked. Okay? Now, did I say anything about this messenger RNA going into the nucleus and modifying your genome of any sort? No. So the Moderna vaccine, in that sense, was a lot safer than if you have a DNA-based type of vaccine. And here's the other example, Johnson & Johnson, right? They use adenovirus to clone a piece of your coronavirus, SARS-CoV-2 protein into this DNA of this virus. DNA was modified, okay? So what happens now? You get the injection, forget about electroporation, just injection, okay? You get the injection, you get fusion of that particle just like it happened over here, okay? And in this case, your DNA is going to be imported, is going to go into the nucleus of the cell. From the nucleus, what is going to happen? This is a piece of DNA, right? You're going to have transcription. You're going to have formation of messenger RNA that is going to come out of the nucleus and make protein in the cytoplasm, and then the story is the same. Okay? So there are certain risks involved with this form of DNA vaccine, because although the company, pharmaceutical company said that the virus is modified and this thing is not going to go and join the rest of your chromosomes, it could be a little risk that maybe that thing would happen. Okay, so it is different perspective here. But now, have you seen now this example of application of nanoparticles and how it can be used, right? So it's not just memorize what is on the PowerPoint, right? And it gets more interesting. All right, so here we get to membrane transport, which you already have talked about it before, but we're going to go into more details on your membrane transport. You can have two forms of transport. All right, guys, let's pay attention here. One is passive, the other one is active. For your passive membrane transport, you are going to rely on diffusion. You're always going to go from area of more concentration to area of low concentration. That's what diffusion is. And you don't need energy for that. You just use diffusion, okay? So there are two ways of doing diffusion. One way is a direct diffusion, that the molecule just bursts through the bilayer and gets in the cell without any help from any protein. That only happens with gases and very, very small hydrophobic molecules. So gases, right? Carbon dioxide, oxygen. Here is also saying carbon monoxide, but that's a poison. That's why it's also so dangerous. It just goes through the membrane, okay? And kills you. And you can add here small hydrophobic molecules, okay? Like steroids. Steroids go straight to the membrane, steroid hormones, and get inside of the cell, okay? All right, so this is direct transport, direct diffusion. Then you have this other form of diffusion that is called facilitated diffusion. That diffusion needs transport proteins. Okay, still you're going to go things that are going from high concentration to low concentration, but now you need a protein to make an opening on the cell membrane, and then the thing can go across, right? Examples are like glucose transporters, and you can have water that goes through aquaporins, okay? Aquaporin channels. Still going from the concentration, more concentration of water to low concentration of water. That is your osmosis, right? All right, so this is your passive, okay? Now, when you look at active process, you're always going to need energy, okay? Because the active process is going to push molecules against their concentration gradient. So in order to do that, you have to put energy into this pump. So classical example is your sodium-potassium pump, in which you have this channel in one form of conformation. Then you bind ATP and you hydrolyze ATP, and the channel changes conformation. And what happens here, it is going to dump 3 sodiums outside of your membrane, and then once it gets rid of the ATP that is bound to the other side, you're going to put 2 potassiums inside of the cell, okay? So sodium has more sodium outside, you're pumping it against the concentration gradient. In potassium, we have more potassium inside, you're also pumping it against your concentration gradient, right? So this is a primary active process, right? When the channel directly uses energy from ATP. On the other hand, you also have secondary active processes, okay? So here, this is your primary active process. But then you have secondary active process in which you make use of the gradient and you push another molecule up this concentration gradient. I think I do have a slide for that. So just in case here, you have your membrane, you have a channel, okay? So in case of secondary active transport, let's say you have here your concentration of sodium that got to be high because of the sodium-potassium pump. And now this channel, let's say it can transport sodium and sugar, so this is glucose, at the same time. So this is going to be a syn-port mechanism that uses your gradient of sodium to sneak in a molecule of sugar that is also going, this is going down the concentration gradient, and this is going up the concentration gradient because you're going to have more sugar inside of the cell. Okay, so this is a form of secondary active transport. It really depended on your sodium potassium pump to get this gradient in the first place. But this should not be the first time that you're seeing that, right? All right. So again, looking at overall process across membranes, if you are looking at the artificial membrane or synthetic membrane, like the book is showing you, right? You see that process, that gases and very small hydrophobic molecules will just diffuse across, but then other small uncharged polar molecules are not really able to pass across. But if you're dealing with a synthetic membrane, eventually you get some of it that goes across. Okay? Now, if you have large unpolar molecules, you even get less of that going across. But again, with the synthetic membrane, some of it is still going to leak through. Okay? But when you're dealing with ions, ions will not go across even a synthetic membrane. Ions cannot go across the membrane at all. Okay? They need a channel for that. So what is going to happen here with this electrochemical gradient, right? We know the sodium-potassium pump is very important at forming the electrochemical gradient, right? And now we need to know what that means. Okay, so when you're talking about electrochemical gradient, you know there's going to be a chemical thing to it, right? So here, let's say your sodium molecules got accumulated outside and they want to go inside of the cell because you have this gradient for diffusion. So that will be your chemical gradient. Now, when you're talking about electrochemical gradient, the thing is different. You also have this gradient that is the same like that, and this thing also wants to go inside. But now you have to look at the electrical charges that are separated at the membrane. So what happens here is that you're going to have more positive charges outside of the cell and less positive charges inside of the cell, and that will lead to a more negative environment inside of the cell. So now you are going to have less positive charge for the gradient, right? And you're also going to have this overall negative charge that is going to pull your positive charge from outside of the cell. So you look at the size of the arrow, your pull is going to be a lot greater when you have a negative membrane potential in the inside of the cell. Okay, so this flushing through the channel is going to be more potent if you put electrical and chemical gradients together. All right. So I already mentioned that. So this is for your study. Okay. So you have your passive diffusion, and that can go through either straight or through cells. And this is exactly what I had mentioned. And this is so you can study, okay? And here is your active transport, and here is what I had mentioned before. So active transport, things are going to be moved against the concentration gradient. You're always going to use energy either directly or indirectly, okay? And what else can make things go against their concentration gradient? you are going to need either ATP or you're going to use light energy. Okay, so these are direct forms of active transport, and here is your indirect form of active transport when you couple it to something that is going downhill. But remember, this gradient was possible because of energy usage in another system, usually your sodium-potassium pump. Okay, so if this is sodium, sodium is going to go downhill. Sodium is indirectly coming from your sodium-potassium pump. So you got to know the distinctions between these forms of membrane transport. All right, so how you control these channel functions? You can have channels that are open, or you can have channels that are closed, and you can use some form of messenger or some kind of chemical that is going to go and open the channel. If that happens, you call the channel ligand-gated channel. Okay. Examples of that, of the ligands, can be neurotransmitters or some of the nucleotides or second messengers. Okay. You can have channels that are going to sense a difference in the voltage. So those are going to be voltage-gated channels. And you still have other channels that are going to feel a change in the shape of the cell. So those are sensors that depend on mechanical stimulation. So you can have this mechanically gated type of channels. Okay. So here again, your sodium-potassium pump. Very, very important. Okay. One main function of the pump is that it doesn't want your cell to burst. So in order for you to keep your osmotic balance, you have to keep the pump active. Okay. All right. So here is a search from your book. I think this channel is looking a bit complicated. What is the main idea that you have to keep in mind? From the cytosol, you're going to have usage of ATP. You're going to have 3 sodiums that are pumped out. And from the outside environment, you're going to pump 2 potassiums in the cell. Gradient that you end up is a lot of sodium out, a lot of potassium in. All right. Consequences of sodium-potassium pump action. Inside of the cell is going to be more negative because you're going to have more positive charges outside of your cell. Okay? Because remember, both sodium and potassium, each one of those ions have 1 positive charge. Pump 3 out, 2 in, you're going to have a charge imbalance. All right, so sodium out, potassium in. And the fourth consequence of the channel is that you're going to have more chloride ions that are going to accumulate outside of the cell. Okay? Because outside of the cell, you're going to have more positive charges. And then your positive charge is going to have that electrochemical gradient to pull your chlorine ions to be outside of the cell. Okay. And another question for you, and yes, relatively easy. Which of the following is an example of active transport? So this one is just your primary active transport. You don't need more than 30 seconds for that. Right, does anybody need more time? No? Everybody got the points? Okay, what is your answer? A. Okay. And here we go. Membrane potential. Did everybody take what do you call it? Anatomy and physiology, neuroscience. Anybody took neuroscience? One person? Anatomy and physiology? A couple of people. Some of you are taking it now. So this is just a review of the test for you. A month. Okay. So what's going on here? Because of that sodium-potassium pump, you are going to have certain resting membrane potentials, right? Depending on the cell type, your resting membrane potential that has to do with how negative the inside of the membrane is, inside of the cell, close to the membrane, right? It can go from negative 20 millivolts to negative 200 millivolts for animal cells, right? And now when you look here at this picture from the book, you'll see that there's one site here that binds potassium, and it says this is potassium and ouabain binding site. What in the world is that ouabain thing? It's a poison that binds to here, binds there as competitive inhibitor, and blocks the functioning of that sodium-potassium pump. Can it kill you? Yes. It can stop sodium-potassium pump. That can kill you. Okay? Again, the problem is in the dosage, right, of this poison. But yeah, this is something that can mess up sodium-potassium pump by blocking the functioning of the channel. Okay? So here. So now we look at electrically excitable cells. So every cell is going to have a resting potential, but some cells are capable of responding to electrical signals, right, or to some kind of signals. And those cells, they have to do with some muscle cells, nerve cells, egg cells or endocrine cells. They can have some kind of response going on, okay? Now, what is going to happen here, you're going to have some voltage-gated or ion-gated channels that are capable of changing your resting membrane potential, okay? So that, of course, is used as a means of communication. And now we're going to get into more details on how you can use the communication via action potential to send a signal and some commands to excitable cells. Okay? So who is familiar with the action potential? Who has seen action potential before? One person, a few people. Now I want to see hands up because that tells me how much you have to study for next exam or not. Okay. So this is review for you, right? And I want to take a couple of minutes to tell you answer to a question that some people have been asking me during my office hours. How can I study more and better for the exam? Okay, how can you do that? You notice that your files for the class, they are all in PDF, right? And everybody should have access to AI, like ChatGPT. Yes, you know how to use it? Who has never used ChatGPT before? If you have never used it, you come and talk to me. Okay? All right. What do? You get your PDF file. Pay attention. This is a lifesaver. Okay? Get your PDF file, dump it into ChatGPT, and you put a prompt with it. You say something like, write 100 multiple choice questions based on this lecture. 50. Depends on how much emphasis you need to put in a particular lecture. Okay? So what is it ChatGPT is going to do? It's going to write the questions based on the file that you gave to it. Right? Oh, also put in your prompt that you don't want to see the answers with the questions. Answers should be placed at the end of the file. Okay? So you go through your questions. The more practice you get answering questions, multiple choice questions, the better you're going to be and more confident you're going to be at the time you come to the class to take your exam in Halloween. Okay? Everybody got that? Okay. So just to remind you how to use it, right? So here, action potential. What is going to happen with your action potential? Something has to come from the outside of the cell to give it a push. That push is going to cause a local depolarization at your membrane. Okay? So the inside of your membrane is going to be less negative than it was before. All right, let's keep going, guys, because I'm going to zoom through this, right? And when that happens, you can generate You have the potential to generate action potential and have a response at that excitable cell. So here it is, right? So what is happening here? This is a thing at your neuromuscular junction. Okay, so here is a muscle, and here is your end of your neuron at the axon. Okay, at the axon, you're going to have a neurotransmitter. It's going to bind to a receptor here. In this case, the receptor is part of the channel. Okay, so this is a bit misleading over there. So let's look here at your action potential. There we go. Okay, you have your neurotransmitter. In this case, is acetylcholine. Remember acetylcholine? Right. At the neuromuscular junction, acetylcholine goes there and binds to the channel that was closed. Once you have bound ligand, this channel, which is a sodium channel, opens. Okay? So this ligand-gated sodium channel opens, and it allows sodium to go inside of the cell. So now you are messing up that resting membrane potential, and you're going to cause local depolarization. When you have a depolarization, local depolarization, so here you know that the inside of the cell is going to be less negative, going towards more positive, right? If that local depolarization triggers opening of another ion channel that allows positive charge to get inside of the cell, that means your inside of the cell is going to become even more positive, right? So the summation of these charges, they are going to increase the excitability of your membrane. That means this signal here is an excitatory signal. Okay, so if this thing is helping the inside to become more negative, you are going to have an excitatory signal. Okay, so overall, this thing is going to be more, is going to be less positive. Okay, now in comparison to something like this, instead of having positive charge going in the cell, if you push in negative charge that goes inside of the cell, now you're going to shift your membrane potential lower than your resting membrane potential. Okay, so what does that mean here? Here is your resting membrane potential. If you add more negative stuff, you're going to go lower than your resting membrane potential. This is going to be more negative. If you have positive thing going in, then you're going to go more to the positive side. So that is excitatory going to the positive, inhibitory going to the negative, which is pretty much intuitive. Okay? So here, what is happening here at the action potential? Right? So let's look here at your neuron and all the information that is going on in your head right now, because you're thinking about moving your foot, but you're not actually yet moving your foot. You're just thinking about it. If you think hard enough and you actually want to move your foot, you will move your foot. So what is happening here, you are sending inputs or information to this neuron that is going to command moving of your foot. But until you reach enough potential at this area here of your neuron that is called the axon hillock, very close to the axon, until you reach enough depolarization there because of this input of information, until then, nothing is going to happen. But you're going to have a certain threshold voltage that if you reach that threshold, then you're going to send your action potential down your axon. Then you cannot stop it. Okay, then it's going to be this all or none type of situation. But the action potential is going to initiate at this area of your axon hillock, and this area inside the cell, it has to reach that threshold. So here is another question. Where is it that you generate your action potential? definitely 30 seconds is more than enough for that. Okay, 5 seconds. Okay, what is your answer? ADC at your axon hillock. Okay. All right, so here comes a part to what is happening to the channels, right? So you can have closed channel. In this case, here is your sodium channel that are voltage gated, okay? So voltage gated means that your sodium channel is going to feel the difference in the voltage, and when they feel the difference at the voltage that has reached the threshold, the channel will open. It's going to stay open for a very short period of time, and then is going to inactivate. Okay? So this sodium channel has a little ball and chain that is going to quickly inactivate the channel once certain time has passed and the voltage inside of the membrane became positive, like positive 30 millivolts or something like that. Okay, so it opens at threshold, then inactivates at positive voltage, and is going to stay inactive until the sodium-potassium pump pumps out enough charge for this thing to get back at the regular closed conformation. Okay, so this is all protein conformation. Okay, so what is going to happen now? When your channel is inactivated, you have this positive charge inside of the cell, and the positive charge is going to bump into other channels that are closed and open those channels. Okay, so the direction of your action potential has to be going down your axon. And one reason for this inactivation, temporary inactivation of these sodium channels, is that if you have a channel that opens and then closes, and then the signal is going this way, and then this channel decides to open right away, you could actually send the signal backwards, and you don't want that to happen. Okay, that would be like short-circuiting of a signal in your brain. Okay, so inactivation of the channel is very important for the direction of your action potential. Okay, and this guy is only the sodium channel. Okay. All right, so this is what I mentioned. All right, so here are the different conformations of your sodium channel that is voltage gated. Here is a closed conformation. Here is an open conformation, and this is your inactivated closed conformation. Okay, so after your channel becomes inactive, this is when your potassium channels are going to open, and you're going to have a flood of potassium outside of the cell. What is they're going to do to your membrane potential? It's going to make it more negative, right? So it's going to start to reestablish your membrane potential by kicking your positive charges out of the cell. Notice that your potassium channels, they do not have that inactivation ball chain. Okay? They do not inactivate. Okay, and they are a bit slower than your sodium channels. Okay, got that. All right. And once you have your signal that went down your axon, you want to reestablish your resting membrane potential, and again your sodium potassium pump is going to come into action and reestablish that. Do we have questions about that? Okay, so interesting things here about your action potential is that when you have your sodium channels that are back to their closed conformation, but your membrane potential is not up to the resting membrane potential, you go through a certain period, actually two periods. The first stage is an absolute refractory period. The second stage is a relative refractory period. You remember this thing about sodium about the action potentials? No. Oh, okay. All right. So here is your time, and this is voltage. Here is your resting membrane potential. You receive a signal, and then you open your sodium. Open the sodium voltage gated channel. You reach a certain positive voltage. At this point, your sodium channel is going to inactivate. And at that point, you're going to open your potassium channel. So your membrane is going to go down. And it's going to shoot past your resting membrane potential. At this stage here, up until the time that you reach that point, there is a period of time here in which your sodium channels are inactive, right? From this time to about this time here, your sodium channels are inactive, so the ball is stuck in there. Okay? This time is what you call your absolute refractory period. If you try to send more signals to your muscles, you cannot contract the muscle again. Okay? Past that time, when you have your sodium channel closed but not inactivated, this is when you reach your relative refractory period. That means if you send a signal strong enough, you are able to send another action potential down this axon. Okay? And why is it you need a stronger signal? Because you are further down at the membrane voltage that is lower than your resting membrane voltage. Okay, so make sure you understand those principles. Okay. When your membrane potential is at lower voltage than your resting, you call this membrane to be hyperpolarized. So that's the hyperpolarized membrane. All right, so here just to reiterate what I just mentioned. Stream, channel, okay. So here back to the resting membrane potential because of your sodium potassium pump. And here we go. Okay, this is a graph that I just had for you. You have absolute refractory period, and then you have your relative refractory period. And here is what is happening with the channels. Now I want to get to another question. Yes, I still have 5 minutes, so 30 seconds here. Grab your question quickly. Grab your points quickly. Action potential is caused by first opening what and then opening what else? Right, everybody has answered. Okay, and what is your answer? A. All right, so I'm going to stop here and pick up from